So we are back with our early career speaker, Marcus Vieria, and he is a senior research scientist in the Kobe lab at the University of Chicago, where he also obtained his PhD. He uses computational, statistical, and mathematical tools to study immunity at different scales, from the within post-evolution of B cells to the effects of post-immune history on epidemiology and evolution of viruses. And today he talks about one of his, I believe, recent papers on germline-encoded specificities and the predictability of the B cell response in models and experiments. Thanks very much for being here, Marcus.
Can everyone hear me? Okay.
Yes.
Thank you, Victor, for the introduction. And thanks to the organizers. I'm really glad to be here. An honor to follow after Hashem's talk. So I want to begin with a shout out to everyone who collaborated in this work. My supervisor, Sarah Kobe, and our amazing collaborators who performed the experiments and sequencing, and who are current or past members of Patrick Wilson's lab. Patrick's now at Cornell, and Scott Boyd's lab at Stanford. And the work is currently out as a preprint, but we will have a revised version soon based on some very thoughtful comments from reviewers. But the sort of deeper motivation for this project is the idea that adaptive immunity evolves on two different timescales. The longer of those timescales is that of the evolution of germline genes that recombine to produce adaptive immune receptors. So in the case of the B cell receptor, the V, D, and J sets of the heavy chain and the V and J sets of the light chain. And those genes have diversified over hundreds of millions of years of evolution in jawed vertebrates. And the shorter of the two timescales is that of the evolution of B cell receptors within the body, which takes place in the days, weeks, and sometimes years after exposure to an antigen. And so we're broadly interested in how these timescales interact, how the long-term evolution of the individual pieces affects their short-term evolution as recombined receptors, and vice versa. As you all know, this short-term evolutionary process involves multiple B cell lineages that are recruited from the naive repertoire into germinal centers. And as those lineages expand, they compete with each other for access to the antigen and undergo selection for mutations that improve affinity. And over time, the lineages that are able to evolve the highest affinities tend to outcompete the rest, even though there's evidence that the process can be quite permissive, in allowing low affinity cells to persist in germinal centers. And so the rate of this dominance can vary between GCs. And then this process is repeated in parallel across multiple germinal centers. And the outcome of it, so which lineages win out, what epitopes they target, and how well they target them, is partly what determines the specificity and potency of the antibody repertoire. So we want to ask to what extent the outcome of this ecological and evolutionary process depends on differences in fitness between the lineages associated with using specific germline genes versus how much it depends on the subsequent evolution of the lineages. And this is related to the broader question of how many evolutionary paths there are to high affinity. So we know affinity maturation can vastly improve binding, yet high affinity for particular epitopes can be hard-coded on individual germline genes from the start. For instance, we know from structural analyses that germline-encoded residues that are present in some germline genes, but not in others, can be critical for binding certain epitopes. Some of these germline-encoded residues are in CDRs 1 and 2, for instance, so they're not affected by VDJ recombination. They're solely encoded by the V gene. And so we might expect B cell lineages using those germline genes to have higher fitness than others, and therefore for those genes to dominate the response. And for some antigens and epitopes, we do find that the response is very strongly dominated by a handful of genes, like in this example from monoclonal antibodies that bind certain epitopes on influenza HA, and they're dominated by a handful of germline V genes. But in other cases, we find that most germline genes are used in the response, like in this example from SARS-CoV-2 antibodies. And the plot is cut off here, but there's actually more than I'm showing. And this suggests that even though some of those genes seem to be inherently better than others and end up overrepresented, the choice of germline gene does not completely determine the outcome of competition between B cell lineages. And so the degree to which specific germline genes dominate the response is part of a broader theme of convergence in the B cell response, which also includes many observations of public CDR3 sequences shared by different individuals. And we still don't fully understand how these convergence patterns arise from the underlying B cell population dynamics. There are several factors that vary between these studies that might affect the extent of that convergence. For instance, the complexity of the antigen, whether we are looking at a single epitope or entire antigens or pathogens, the type of B cell we're looking at. But one fundamental dimension we thought was unexplored is time. So how the degree of convergence in germline gene usage changes over time as the extent of affinity maturation increases, and so does the opportunity for mutations to overcome germline-encoded fitness differences. Oops. And so we decided to tackle this problem using a combination of simulations of B cell evolution and competition and sequence analyses of the mouse response to flu infection. Underlying both the simulations and the experiments is an approach to thinking about and quantifying the relative importance of what I'm calling these germline-encoded fitness differences. And from this point on, I'll use gene and allele sort of interchangeably, just because the argument applies both to different V segments occupying different loci in a single chromosome and to different versions of a segment occupying the same locus in different chromosomes. But as I mentioned before, these fitness differences might arise if naive B cells have different affinity distributions depending on what germline allele they use, for instance, depending on the heavy chain V allele. And affinity is a property of the entire receptor, of course, not of its individual pieces. So for any given heavy chain V allele, and I'm representing here 3 in different colors, we have a distribution of naive affinities depending on what alleles are used in the other sets. So this variation here comes from the D and J genes in the heavy chain and the V and J in the light chain, and also insertions and deletions, etc. But the point is that those distributions might be different between alleles, so that some germline alleles have a higher propensity than others to generate good receptors against a particular antigen or epitope. Another possibility is that receptors might have different adaptability distributions. So the propensity of a naive B cell to adapt via affinity maturation might be affected by its germline V gene. For instance, if V genes have different somatic hypermutation rates or different fitness landscapes with respect to a particular antigen. So this is another way in which fitness, in this case, sort of fitness in the long term, might depend on the choice of germline allele. And if these germline-encoded fitness differences strongly affect the outcome of B cell evolution and competition, then we might expect specific alleles to be overrepresented in the response to the antigen relative to their frequency in the naive repertoire. So here I'm showing naive frequency on the x-axis and the frequency in the response in the y-axis. And what's more, to the extent that different individuals have similar sets of germline alleles, though they're usually not identical, and their naive frequencies and recombination probabilities are similar, i.e. these distributions are similar, we expect the same alleles to be overrepresented in the response of different individuals exposed to the same antigen. So not only allele frequencies in the response, but also their deviations from the naive repertoire would be correlated between individuals if fitness strongly depends on the choice of germline allele. But now consider a different scenario where fitness differences mostly arise not from the initial choice of germline allele, but from during the subsequent evolution and competition of the lineages. These processes are subject to different kinds of stochasticity, so which lineages win out may depend on factors that are largely random with respect to the choice of germline allele. For instance, it might depend on when different lineages arrive in the germinal center, on the timing, order, and effect of mutations in different lineages, or demographic stochasticity and genetic drift. And whatever germline allele happens to be used by the lineages that sort of win this lottery, those alleles will tend to increase in frequency relative to the naive repertoire. But since those stochastic processes play out differently in different individuals, there would be no tendency for the same alleles to be overrepresented in different individuals. And so both allele frequencies in the response and their deviations from the naive repertoire would become uncorrelated between individuals. And so we can look for these correlations between individuals as a way to measure the strength of germline-encoded fitness differences. And we can ask whether one or the other scenario is more likely at different times during the response. So to formalize these ideas, we turned to a model, a simulation model of B-cell competition and evolution under selection for affinity, in which each naive B-cell has an initial affinity drawn from a distribution associated with its heavy chain V allele. And the mean of this distribution is allowed to vary between alleles. Affinity in the model affects recruitment into germinal centers and rates of cell division. And so combined with somatic hypermutation, this leads to affinity maturation. We assume no migration between germinal centers. And finally, we based the simulations on empirically determined germline V sets of black 6 mice. I'll talk more about these experiments in a second, but for now, I'll just note that those alleles have similar frequencies in the naive repertoire of different mice. And so we first considered a scenario in which all V alleles are functionally identical. They have the same naive affinity distribution. They're basically just labels. And just as a sanity check, we see that over time, germinal centers become increasingly dominated by a single lineage that evolves the highest affinity. So here we have the frequency of the biggest lineage within GCs as a function of time, and the different colors are different mutation rates. So the higher the mutation rate, the faster this dominance by a single lineage tends to happen. But then we can look at the correlation in germline allele frequencies between individuals over time in their response. And note that in this plot, the colors represent the number of germinal centers per individual and not the mutation rate. And so in this baseline scenario where alleles are functionally identical, germline allele frequencies are correlated between individuals early in the response. And that's because B cells are being recruited from the naive repertoires, and the naive repertoires are similar between individuals. But then the correlation between individuals decreases over time. And what happens is that without differences between the alleles, the probability that two GCs are dominated by the same allele is low. And so with a limited number of germinal centers per individual, the set of winner alleles is different in different individuals. And then as expected, since all the alleles are functionally equivalent, there's no tendency for the same alleles to increase in frequency relative to the naive repertoire in different individuals. So there's no correlation between individuals in alleles experienced to naive frequency ratios, which is how we are measuring these deviations. And then next, we simulated what happens when the alleles are not all identical, when 5 V alleles have higher expected affinity than the rest. So 5 alleles look like the orange allele in this schematic, and the rest are all identical, and they have identical affinity distributions. In this scenario, 2 critical factors that determine the degree of similarity between individuals are the fitness advantage of the high affinity alleles, which is just how much higher their average affinity is relative to the other alleles, and the effect size of somatic hypermutation and the rate of somatic hypermutation, which I'm going to show in a second. So if we start by keeping the effect size of somatic hypermutation relatively small and increase the advantage of the high affinity alleles, we see that the experience to naive ratios stay persistently positively correlated between individuals over time. That is, the same alleles are consistently overrepresented relative to the naive repertoire throughout the response. And the higher the advantage of using those specific alleles, the more consistently those alleles tend to be overrepresented, and so the higher this correlation. And then as we increase the effect size of somatic hypermutation, the correlation starts to decrease over the course of the response. And the faster the mutation rate, and that's shown here by darker colors, the faster this decrease happens. In other words, high affinity alleles are consistently overrepresented early on, but less so over time. And so to understand what's going on in this last case, this plot shows the combined frequency of those high affinity alleles within a germinal center. So one means completely dominated by high affinity alleles, and the y-axis shows the distribution of germinal centers along the x-axis. If we switch off somatic hypermutation, we see that germinal centers are consistently dominated late in the response by lineages using those high affinity alleles. So the GCs stay on the right of the x-axis. And this makes sense because without mutation, those lineages always have higher fitness on average than lineages using the other alleles. And as a result, frequency deviations from naive repertoire stay correlated between individuals over time. But if mutations are sufficiently frequent and have a sufficiently large effect, some lineages using the low affinity alleles will overcome their disadvantage and end up dominating germinal centers. So we see many germinal centers on the left part of the x-axis. And which germline alleles those lineages happen to use varies between individuals because it depends on chance events. It depends on the timing and effect of mutations in different lineages in different individuals. And so the correlation between individuals then decreases over time. So we could summarize this sort of exploration of simulated patterns in this schematic. So if the affinity distributions of naive B cells are the same across all alleles, if there's no fitness advantage associated with using specific germline alleles, then no allele should be consistently overrepresented in different individuals. And similarity in germline usage between individuals will decay over time if there's a limited number of germinal centers. If some germline alleles have higher affinity on average than others, then how similar the response will be across individuals depends on the size of that fitness advantage relative to the rate and the effect size of somatic mutations. If the advantage is strong enough that it's hard to overcome with somatic hypermutation, the high affinity alleles will be persistently overrepresented in different individuals, and so the individuals will have persistently similar germline usage. Oops, sorry. If the initial germline-encoded advantage is small enough that it can be easily overcome by somatic hypermutation, then similarity between individuals will peak early on and then decrease over time. Now, I'll mention that there are other dimensions we haven't yet explored. For instance, the number of germline alleles in these different classes, low affinity, high affinity, and of the parameter regimes that we've explored, different regimes might be plausible in different systems. For instance, it's unclear what the distributions of naive affinity and mutational effects look like for different antigens and germline alleles in different species. But we decided to compare these simulated patterns with the observed response to influenza infection in mice. And so in collaboration with the Wilson and Boyd labs, we sequenced B cell receptors from mice infected with an influenza virus once or twice and then sacrificed at different time points. And we looked at germline heavy chain VLEO frequencies in naive and experienced cells. And we focused on experienced cells isolated from the mediastinal lymph node because, unlike in other tissues, those cells were largely absent from control mice. And this suggested that many of them might be flu-specific. And even though these mice are highly inbred, we found similar but not completely identical germline V-sets in different mice. So here I'm showing the observed numbers as points. Each point and curve is a single mouse, and the rarefaction curves just show the number of V alleles we'd expect to find if we were only sample a subset of the mouse's sequences at random. And the fact that these curves are largely flat suggests this variation in the number of V's across mice isn't due to sequencing depth. And so, motivated by some comments from reviewers, we tested alternative annotation tools and germline reference sets, and we still found this variation. And we haven't been able to find papers systematically, investigating variation in germline sets across individual mice in these highly inbred strains. So we're not sure how surprising this really is. So it's a point I especially welcome feedback on. But then we went ahead and looked at the correlation in germline VLEO frequencies between the lymph node B cells of different mice. So here I have the different days after primary infection, including the controls. On the y-axis, the correlation in heavy chain VLEO frequencies between individuals. And we found that for germinal center cells and plasma cells, allele frequencies were similar between mice, really in the response, but less so over time. So each point here is a pair of mice sacrificed at the same time point. And we also found that frequency deviations from the naive repertoire were often positively correlated between mice, suggesting that some alleles had a higher propensity than others to generate receptors good at binding flu. But in plasma cells, which might be the population most strongly reflective of selection for increased affinity, we found that this correlation dropped very sharply over time, suggesting that specific V alleles were consistently overrepresented early on, but less so over time. And this is just an illustration of this pattern. So each panel here is a mouse sacrificed 8 days after the infection. The x-axis shows the 20 most common germline V alleles in lymph node plasma cells. And the arrow goes from the alleles frequency in the naive repertoire to the frequency in lymph node plasma cells in that mouse. And the deviations are colored by whether they're significant based on bootstrapping. So notice how there's this germline V allele, 14.4, that increased in frequency relative to the naive repertoire in all mice, and it became either the most abundant or the second most abundant V allele in lymph node plasma cells. And later on, and this is not shown in this figure, the most common V alleles and these patterns of overrepresentation become much more variable between mice, and that's what's reflected in this plot. Hang on. So even though we focus on germline gene usage, we also asked whether these seemingly flu-induced lymph node populations had CDR3s that were more similar than the CDR3s of naive sequences. So here the x-axis shows time since the primary flu infection. The y-axis shows similarity, either sequence similarity or biochemical similarity for pairs of sequences sampled from different mice and matched for CDR3 length. And the columns show different cell types. So with naive cells on the left and the three next columns showing experienced cells from lymph node. And then we found that the CDR3s of these experienced cells were no more similar than the CDR3s of randomly sampled naive B cells, suggesting that the flu infection did not strongly select for specific CDR3 sequences. And so these results suggest that germline-encoded specificities shaped the early mouse response to flu, but do not predict the long-term fitness of B cell lineages. So if we go back to the schematic summarizing our simulations, the results suggest that the mouse response to flu might be somewhere in this region, where some V alleles encode receptors with higher affinity than others, but that advantage is small enough that it can be easily overcome by somatic hypermutation during the course of the response. Now, flu doesn't normally infect mice, so any propensity for specific alleles to make particularly good receptors against flu is likely not the product of long-term selection on germline genes. Instead, germline-encoded specificities for antigens that have never been encountered might arise as evolutionary spandrels, so byproducts of the diversification of germline genes, as Maya Sangersland and Daniel Langwood suggested in a recent paper. And so depending on the fitness costs imposed by the pathogen on the host, selection on germline genes might then reinforce those specificities if they help reduce that cost. So perhaps in the response to pathogens that share a long evolutionary history with the host, we might encounter stronger germline-encoded specificities that lead to persistent similarity in the response across individuals, which I think is an interesting hypothesis that takes us back to this initial idea of connecting the long-term evolution of immunoglobulin alleles and their short-term evolution as recombined receptors. And with that, I'm going to thank you all for listening, and I will be happy to take any questions.
Thanks, Marcus. Fascinating talk. And it's one of my favorite things to talk about. Why do we have the germline gene alleles that we have, and how have they evolved, etc. So, but maybe Felix has first a question, and then I ask a question after. No?
Oh, yeah. I mean, interesting talk. Yeah.
Thank you.
How did you sequence the germline genes again?
So we did RNA sequencing on these different. So we sorted them by tissue and cell type, and then performed RNA sequencing.
Sorry. It sounded like you were saying your inbred mouse strain had surprising variability in germline genes.
That's what we found. Yes. Let me go back to that slide.
And but what was like sequencing just the V gene region with primers, or that was doing a pull down? You know, how did those germline genes get sequenced that showed these variability?
It was sequencing of the V region plus a little bit of the C region. I apologize. I really don't know much about the details of sequencing.
Oh, okay. Because I'm just thinking, because that region is so complicated. Sometimes you might find this variability just.
But sorry, just to complement, there were constant region primers.
Okay. Yeah, I was just wondering where that was coming. That's really interesting. I mean, people have been wondering about that. You know, that these inbred strains kept in different places, maybe showing some variability. Yeah.
Yeah, but that's across strains. But they used mice from one strain. So even if they make an error,
one strain
for the same. Yeah. So it's strange that there is variability across mice. I think that's the problem. Yeah.
That's not due to variation in sequencing depth, right?
Yeah.
And so I'm very curious to see if there are any papers that sort of systematically look at that using different annotation tools. But so far I haven't found any. I'd love to hear about that.
But so and you found high variation in the V gene usage in the naive mice, right? Is that what you also found, or if you go to mice?
Including the controls, is that the uninfected mice, right? That's the green lines. Yes.
Maybe the next, but the next the box plot. So the control, I know what is, yeah, okay, no, yeah.
Okay.
So if you go back again to the other, what's the number of genes? Yeah, but what was the correlation of the V gene usage for your naive repertoire?
For the naive repertoire, I think I have an extra slide here. So it was lower than in other papers, including I think the paper that you wrote, right?
Yes.
And so as a sort of sensitivity or analyses, we repeated the analysis of frequency deviations from the naive repertoire using naive frequencies from that paper, which is also based on black 6 mice, right? And we found similar results. I don't think I have the slide here. But I will point out that the gating strategies were different, right? So for calling naive B cells in those two studies, so that might be that our naive repertoire isn't totally purely naive, and we have a bunch of filters in place to remove sequences that were from large clonal expansions or that had too many mutations, that were sorted as naive but had too many mutations in the V gene and so on.
Because also even from human, we always see that it's very high. So it's not even mice. So when I look at sequencing data, I always see that it's very high. Yeah.
Mm-hmm.
Yeah. So it's not only a mouse. So yeah. Yeah, that's who knows. But Ken has a question.
Hi, Marcos. Great talk.
Hey, Ken. Thanks.
A couple of questions. One was on the Partis and IG blast annotation comparisons. Do you know why those look so different compared to between Partis and IG blast? Is it because Partis is inferring some sort of genotype arrangement?
That's a good question. We haven't, so that's a recent analysis. I haven't dug into that too deeply. Yeah, so it looks like IG blast is allowing itself to potentially find a lot of V genes, right? Like it's giving it access to like the entire INGT mouse reference set. And Partis, even when allowed to do that, is much more parsimonious, right? And it gives very similar results when you just use the reference set, the OGRDB curated reference set. Yeah, I'll mention that this is also not due to so Partis by default looks for new alleles, right? That are not in the reference set, but that doesn't change the conclusions in this case, and the variation still persists whether or not you allow it to do that. But I don't know yet why IG blast behaves this way.
Okay. Yeah, I think Partis might infer like the genotype of the mice, so it prevents it from adding too many alleles.
Right. Yeah. It has the explicit goal of not. I think I don't remember exactly how it does that, right? But not of limiting the set, right? Of not using everything that's in the reference set.
The other question I had was, how do you expect these to be different in humans, since mice repertoires tend to be more germline focused?
Right. So for humans, we might see perhaps humans are less inbred, right? So we might see less similarity, right? At least if we were looking at a pathogen with which humans don't share a long evolutionary history, like flu in mice, right? So I might expect similarity to decay faster, because we're going to have more variation between individuals, presumably in the set of germline genes. And as you were saying, the diversity introduced during VDJ recombination is likely higher, right? Does that make sense?
Yep. Cool. Thanks.
Awesome.
Thank you.
Yeah. Hey, Marcus. A fascinating talk. Thank you for sharing. And also, I'm not a B cell expert, so perhaps my question is a little bit naive, and I apologize for that. My question is kind of related to Kenneth's last question. I was wondering how basically your model will explain the differential gene usage in chronic versus acute infections, for example, in humans.
That's a very interesting question, and something that we sort of speculate a little bit on in the discussion. So chronic infections, to the extent that they involve recruiting the same lineages over and over into GCs, they would have a bigger opportunity for mutations to disrupt these initial germline-encoded advantages, right? So it might be that we'd see greater divergence in the response to chronic infections than we do in short acute infections. because there would be more opportunity for mutations to overcome those initial germline encoded differences, right? So that's, I think, largely remains to be tested. I don't know if people have systematically looked at that. But that's one expectation. Of course, this all depends on the extent to which the response to chronic or repeated infections, right? And the argument is similar for repeated infections like flu in humans. The extent to which, as the infection goes on, or with each repeated infection, the response relies on reactivation and continued evolution of memory cells.
Thank you.
Thank you.
Do you want to ask one more question? No, Felix is, yeah.
Well, so I mean, I guess the alternative hypothesis, I mean, to what you saw, in your experiment, would be that there were just some V genes in that mouse strain that were just so good at binding to the antigen that it would overcome somatic hypermutation. I mean, that was kind of the other extreme possible result, I guess. I mean,
to explain this peak early on. And...
No. I'm thinking just, you did your experiment and you see somatic hypermutation kind of overcoming the initial correlation, but I mean, that's the alternative possibility before you did your experiment would be that there'd be just a few V genes that were just so good at binding to the antigen that they'd never, so somatic hypermutation would never overcome those, And
in which case the correlations sort of stay high, right?
Yeah.
Yeah.
I mean, it's it's a funny thing because in evolutionary biology, there's a phenotypic gambit, that mutation can overcome any sort of genetic constraint that genetics gives the organism, even if there's a selective type that it is evolving towards, and it's a very similar argument,
And again, it might be that with more time, right? So, for instance, in a chronic response, that would happen, right?
Well, yeah, I mean, with a whole bunch of antigens and a whole bunch of variability in haplotype between individuals, it's a different story.
And we also think this might depend on, again, the sort of scale that you're looking at, right? If you're looking at a single epitope, then it might very well be that the only way to bind this really well is this specific gene. But then if you're looking at a whole pathogen, right, then it might be that no single gene is that amazing compared to the others when you're considering the entire response against all possible antigens and epitopes.
So there is one question in the chat. Do you see the question, Marcus?
I see it. Is frequency a comprehensive metric to quantify similarity of naive immune repertoires? Or would you think we could complement this with additional metrics to understand better this similarity between individuals? Yeah, that's a great question. I would say that it's definitely the easiest thing we can get from sequencing, right? Ideally, we would also have affinity and we would know, what these lineages are binding. Similarity in frequency might be very deceptive, right? In the sense that perhaps the frequencies are very different, but the epitopes being bound are very similar, right? And so the functional consequences are very different or very similar. So I don't know if I've answered your question, but yeah, I think frequency is convenient, but not ideal.
Thank you very much. I think you have answered my question, and yeah, sorry for previously. So.
Oh, not at all. Thank you.
I have also one more question, and I think then we need to wrap up. How do your simulations relate to previous work or recent work by Corey Watson's lab, where they show that germline gene alleles can bias another allele, a completely different allele? So the usage of it, so this long range effects.
Did you say Alex Walczyk's?
Corey Watson.
Corey Watson, sorry.
Watson, yeah. So treatment a few months ago where he shows that given polymorphisms can bias the usage of another allele, for example. Yeah. So
That's a good question. I have to look into that.
yeah, so it's kind of a long range effect between allele and their usage. So it's not only that the usage of one allele goes up, it's if one goes up, then they can move others up also with them, et cetera, et cetera.
And that's beyond just the fact that these are compositional data. So that, you know, if the frequency of one goes up, the frequency of the others have to go down. It's something, it's more than that.
Yeah, at least I think so.
I will check out that paper. Yeah, thank you.
Yeah, I think there would be, yeah. I'm not sure how one would model that, but it might be nice to take into account. Yeah. And then one last question. So what do we learn from your work? You had it also on your last slide about the long-term evolution of germline gene repertoires. So why do we have the germline gene repertoires that we currently have and not others with respect to the pathogen universe or the antigen universe, et cetera, et cetera? So what?
Right.
Yeah.
That's related to the question, and maybe just a different way of posing it, right? Of how often these germline-encoded specificities are themselves the product of selection on the germline in the long term, right? So I'm inclined to agree with this paper, right? In the suggestion that you're going to sort of get them for free as V genes diversify, because it would be highly unlikely, I think, for them all to have the same naive affinity distribution against any particular antigen, right? The question then is, in what conditions do they provide a fitness benefit that can be reinforced, right? So if it's true that the situation with mice and flu is representative of these evolutionary spandrels, right? So we don't share an evolutionary history, so that advantage, you know, is likely going to be small enough that it can be overcome by somatic mutation. Then the fitness benefit of that germline-encoded specificity might be related to the early response, right? So it might make a difference that you're able to quickly mount, you know, a pretty decent plasmablast response, and if that helps you survive. For instance, if it's a pathogen that you encounter very often, if you're a small species that don't live long, then that might make a difference. And then, you know, then that might, you know, be conducive to selection to reinforce that initial germline-encoded specificity. And that would depend on many things about the life history of the species, right? And also the fitness and cost imposed by the pathogen. If it's a pathogen that you're very unlikely to see, then they may be, you know, maybe there won't be much of a selection pressure to reinforce that.
And I guess
those germline-encoded specificities. Yeah.
And I guess what I think we always forget a bit that yes, the germline genes themselves, but they're also, of course, in VDJ recombination, they make CDRs. And so you haven't seen any connection there, right? As you said, but we only for now have a way to look at the sequence diversity or something. But what I think actually wanted to look at is the functional space of the CDRs, and maybe there we would see an enrichment if we could map every sequence.
So their sequences might not be similar, but something about their shape or function might
be.
And this very crude measure of biochemical similarity that we use wouldn't capture that. It would have to be something for sure. Yeah, absolutely.
Yeah. So I think that's so then the even more further question would be, how is VDJ recombination selected on or the CDR how are the holes in the repertoire by the V gene D and J, etc.
Yeah, absolutely. Yeah, so it's not just the set of individual pieces and its evolution that matters, it's also the evolution of how these things are recombined with.
Yes, that's much better phrased. Yes.
So
okay, I think if there are no more questions, it was great to have you all here today for the first AIRR-C seminar series. The next one is May 25th with speaker from Norway and a speaker from Israel. So looking forward to that. Thanks so much, everyone. Bye-bye.
Thank you.
And thanks, Marcus, and thanks, Hashem. Again, bye-bye.